So the first part of this unit is all about photosynthesis and respiration. Now this is about enzymes. Enzymes are the things that run photosynthesis and respiration because they run all the chemical reactions in all organisms. So these are going to be the enzyme notes. So first, what are enzymes? Well, they're proteins, and since their proteins are made up of amino acids, you know all those circles put together, those amino acids, so those amino acids all put together make up a protein. So to fill these out, so enzymes are proteins, and they're made up of amino acids. So now what do they do? Well, what they do is they'll either, so if they're enzymes in our stomach, they'll digest things and break them apart, and or they'll put two things together. So they can also take these products and put them together to make something. So like if we're building up muscle, they'll build up our proteins and make muscles. And what they do in this situation is they actually help speed up this reaction that would normally occur. So you would eat some spaghetti and it would slowly break down in your stomach, but without enzymes it would go way too slow and you would never get energy from them. So this helps speed it up so you get the energy from the spaghetti quicker. So they can either break down a substrate or put two substrates together. And enzymes speed up reactions. So now we're going to talk about enzyme factors and what's going to affect how the enzyme works, the speed at which it works. So the first one we're going to talk about is temperature. So there's three of them. There's temperature, pH, and substrate concentration. And all enzymes are affected by these because enzymes have to be a specific shape to fit with the substrate. And these, except for the last one, the third one doesn't do this, but if it changes the shape, the enzyme will slow down. So the first one that might change the shape is temperature. So all enzymes work at one specific temperature, and this is called the optimum temperature. If you ever heard of Optimus Prime is the best transformer. Optimum means the best temperature. So work best at one temperature, and it's called the optimum temperature. Now it's going to be different. This optimum temperature is going to be different for different enzymes. Some enzymes are going to work best at all temperatures. It just depends on what organism it is. So this graph is going to look like this. It's usually going to be at the best at one specific temperature, and then if you get warmer it slows down, and if it gets colder it slows down. So our graph, if you take this, if you're working on this Google Documents, if you just click the line tool, and we actually went the scribble line tool, so we'll just take this up and go like this. All right, and then what I'm going to do is I'm going to indicate where the optimum temperature is. So optimum temp is basically, if I take another line here, is right here. Oop. Alright, let's try this again. So the optimum temperature would be right here where it reaches the peak. So let's say if that is, I don't know, 40 degrees or whatever temperature is right here, that would be the optimum temperature. So enzymes in our body work best at, so what temperature does our body have to be? That's 98 degrees Fahrenheit. All right, and that is why it's dangerous if we get too hot or cold. So if we run a fever or if we get hypothermia, we can die because our enzymes will no longer work at those temperatures and we won't be able to do any reactions. The reason why it doesn't work as well at colder temperatures is because the substrates slow down, so they don't bump into the enzyme as quickly, so it slows down. And the reason why it doesn't work at high temperatures is because the enzyme starts to change shape, so it doesn't react as fast. Why this is important, so we do use this concept every single day, even when you don't think about it. So we put foods in the refrigerator, and that slows down the enzymes. so they do not rot as fastly, quick, so they last longer. And then we also heat up enzymes to kill, heat up food to kill the enzyme of bacteria that might make us sick. So like chicken, if you eat raw chicken, you could get sick from bacteria in there. But if you cook it, it kills the bacteria because it kills those enzymes in the bacteria. All right, so that's temperature. So temperature works best at one temperature. In case in our bodies it's 98 degrees. And then either way, it will slow down. All right, so now pH. pH is very similar. So enzymes work at one specific pH called the optimum pH, just like temperatures. So this should be pH. I forgot to change those when I copied and paste it. So at one pH called the optimum pH. So our graph with the optimum pH would just be like our temperature. If I draw a scribble here, makes that little parabola there, and our best pH would be right there. All right, so let's say if this was going to be a pH of 7, it would work best at 7, or like water is. And then remember, our pH scale this way it gets more acidic, and this way it gets more basic. So that's how pH affects enzymes. And each enzyme works best at the pH for the environment it is in. So the pHs in our stomach would work very best in a low pH. So pepsin's in our stomach, it works very best at 2 because our stomach is very acidic. Whereas the pH of trypsin works best at a pH of 8 because it occurs in things like our intestines. So if you make pickles, you put pickles in vinegar, and vinegar has a very low pH, so it slows down enzymes, and that's why pickles can last very long after you buy them, because they have that low pH. Alright, so now the last one is substrate concentration. So substrate concentration, the more substrate will lead to faster reactions because there's more substrate to bind to it until the enzyme cannot go any faster. Alright, so once the enzyme can't go any faster, it will level off. So the substrate bumps into the enzyme, really makes it go quick, fast, but then eventually the enzyme can only go so fast. So it's kind of like your car, anything, you can increase, but eventually you can only go so fast. There's a point of where no matter what you do, you're not going to get any faster. So you just level off. All right, and that's same here. It's because the enzyme can just no longer go any faster. So you can put more and more substrate in there, but it's not going to go any faster. So we're going to show that on our graph here. So again, I'm going to use the little scribble line here once it loads. Come on. Alright, and then our enzyme speeds up, and then it starts to level off. Alright, so it just starts to slow down, and it doesn't increase anymore with any more substrate. Alright, so last thing is now if we put on all those factors together, their graphs would look like the substrate curve, but then we can put different lines on there for different temperatures and different pHs. So this shows again that as we increase substrate, the enzyme level off. But now you see this one is very fast, this one's medium, and this one's very slow. So we'll say this one is when the enzyme's at its optimum temperature because it's the best one. And then this one would be if the enzyme was heated up or cooled off. And this one is where it's very cold or very hot. It's very, very slow. All right, because it just slowed down. Same with pH. This one would be the one at the best pH. This one's more acidic or basic, and this one's very acidic or basic, and so it's not going to work as well. So if we, so the graph shows that the enzyme rate after the substrate gets so high, so the enzyme rate increases. What's going on here? Increases after the substrate concentration goes high, and then levels off. And then the higher the line, the closer the enzyme is to the optimum. So the higher the line, the closer is the optimum temperature or pH. So the higher the line, the closer it is to that optimum. So if we did that again, so again we'll take our scribbles here. So the optimum temperature temperature or pH line would look something like this. So remember, it levels off because the enzymes can only go so fast, but it's really high. And then this one is kind of close to the optimum temperatures or pH, whichever one we're doing, but it's a little bit slower, so it's not quite the optimal. And then this one is not even close to optimum temperature or pH. It's very slow. So this is the best, and then this would be the worst. So if you put that one, so this would be the optimum temp slash pH, and then this would be the, down here would be the worst pH or temp for the enzyme. Alright, so again, the enzyme just levels off and can't go any faster, and then this is how temperature or pH affects it. It will cause it to go down or decrease. Alright, and that's it for these notes. So enzymes are very important because they speed up reactions. If we didn't have them, we wouldn't live. And then we work with them every day because you get fevers, you use a fridge and a freezer, so you're constantly slowing down and speeding up reactions without even thinking about it.